required to understand the research. What are the fundamentals of clinical research and how to design experimental research. The second part is almost a workshop, and I will give you assignments during my lectures and during my presentation, and you will solve it, and then I will give the answers. There are mainly two types of researches in our medicine and in medical field. One is called the clinical and the other is called the basics. Mostly during our thesis or during our research in basic sciences, we do not conduct the clinical research, but we usually, basically, we do the clinical research. The clinical research involves the scientific investigation of the etiology, prevention, diagnosis, treatment of the human disease using human subjects, human population are the materials. While the basic research that we usually do basic research or the fundamental research is a type of investigation focused on improving the understanding of the particular phenomena that has already been, to study the DNA, to study some brain function, to study the heart function, to study the cardiac function, to study the function of the enzyme, to study these all things, to study the law of nature that is already there in our system that's based in the basics. So these two are differences between the researches in MPhil and PhD. When we are doing the basic sciences, mostly we conduct the basic research. But it does not mean the basic research means the research in the basic sciences. No. You can do even the clinical research in the basic sciences, and you can do even the basic research in the clinical sciences. Let's see what are other important things. How we can do the research. There are so many types of research. There are so many types of the classification of research and the present the literature, present in the Google. So I will try to solve to make them easy to understand you all peoples. So suppose first of all, we think in our mind when we are starting the research, is our research needs intervention? If the answer is no, there is no intervention in our research, or there is yes, there is intervention. If there is no intervention, simply the research will be your observational research. And if there is intervention, either injection or either medicine or either your instruments or something else, then it will call an experimental. It could be animal experimental or it could be human experimental. And if observational, then if there is no analysis, there is no analytical methods involved, simply you are going to draw the inference, not drawing the inference, then it is called the descriptive study, descriptive research. But if some analytical processes are there, that is comparative or analytical methods are involved, no doubt it is observational, and then it is called analytical study in analytical research. Descriptive research, we usually start at the student level in first year, second year, third year, fourth year, final MBBS students. And whenever you see any important case in your life during your residency or during your practice, you do it. You simply describe the case report. Sometimes there are more than one reports, and then it's called the case series. Even you can do the survey during this COVID if you want to ask the question of your answer, how many people have taken the vaccine. You can, this is called a survey. So survey is also a descriptive study. But when there is analysis in this descriptive study, case report and case series, you do not need to include any inferential methods. You do not need to make any hypothesis or something else. But when you want to draw something that is cross-sectional study, once again, the cross-sectional study, we have to see there is prevalence, there is incidence, something else. Another study in this area and the observational when there is no intervention, that is a case control series, and then another big research that is called the cohort. On the other hand, when it is experimental, when it is animal, it is called animal study. When it is human, it is randomized control. Randomized control are once again, if there is quasi, there is control or not control, there is something, one parameter missing, that is quasi, or if this yes, then it is called the randomized. There is another way of this classification beautifully, that if you assigned the investigation, yes or no. If yes, then it is experimental study, randomized allocation, yes or no. If there is no, then it's called non-randomized trial or quasi-trial studies. I have given the detail of same observational, but at the end of this, the cohort study, when there is exposure, you want to see the outcome. Suppose there is an exposure after COVID somewhere and you want to see the mortality. Or if there is outcome, there is a mortality, you want to see the cause of this, then it's called the case control study, or this. The same as the cross-section studies. There's some other terminology that we usually use. What is exposure? What is outcome? Exposure mean environmental, or as I have seen in this case, exposure and outcome. So what is the exposure means? Environmental or clinical factors that could potentially influence incidence in certain diseases, mortality, like smoking, like alcohol, like COVID, like viruses, like smoking, that slow birth child. So these are the exposures. Outcome means when smoking, you want to see cardiovascular disease. In the smoking, you want to see the association of cancers, lung cancer, same. That is count. So these two terminologies are very important to see what is the exposure and what is the, these are the two important fundamental things to understand before starting your research or starting this. There are so many other classification of this study: observational, descriptive, analysis, cohort. These are different types of the controlled and uncontrolled studies. Hierarchy is the same, observation. So there are certain other terminology that we use. Retrospective means backwards and prospective means forward. The study involves collecting data about events that have happened before already. Suppose the COVID was in 2019 and you want to study the effect of COVID in cardiovascular system, you will do in the retrospective study. And the prospective study, now the COVID is almost over, but you want to see the post-COVID syndrome. You want to see the data, and that post-COVID syndrome, that will be your prospective studies. What is going to be happened in the future. Randomized means the important study, that interventional study, very, very important. So you have to understand what is the randomization. I'm not going to make you the detail, but a very simply terminology, fundamental terminology that I will define in my lecture, that randomization mean a randomized controlled trial is a type of scientific, clinical study, medical study, experimental, which aims to reduce the bias when testing a new treatment. No doubt there are so many bias. The actual study is to reduce the bias, but important drawback of the study is that there are so many bias. To reduce the bias, it is a very important thing to make a good questionnaire. Very good questionnaire will reduce the bias study, because honesty is the best policy, and I think the honesty should be the basic thing in the research. That is why mostly in countries, the countries are not progressing, because they are not doing the research where they are honest. There are so many biased. The people participating in the trial are randomly allocated. Everybody has chance to participate in it. To either groups are receiving the treatment under investigation, or to group receiving the standard treatment, placebo, which is called the treatment. This is called the randomized. When I will give you, there are three important things. What are the three important things required for the randomization? So randomized trial, randomized control, there are two things: randomization and controlled. Randomization, when you are doing something, giving some people are receiving something, treatment, and control, which are not receiving any treatment. In the trial, the drugs or the treatment on the trial during this research study. So this is the chart that's showing an experimental on one side and the control on the one side. A very important question, that it does not mean if you have 10 people in experiment, it does not mean you have 10 people in control. You can get 9, you get 11. If there are certain control studies which are called age matched, gender matched, or so if there is some matching, then the both group have similarities. Otherwise, the both group do not have the similarities. So there is some specific time we have to use these things. So this chart is showing the randomization, experimental or the controls comparison, first data collections, period of intervention, last data collections. Sometime in the trial, you use the pilot study on the few subjects. And after getting the results, after comparing the results, you start another study. So this is very, very important, that what are the important features, what are the important fundamentals things in randomization. There are three things. That is called I for interventional. If there is no intervention, there is no randomization. If there is control, or if there is no control or comparative, then there is no, and the randomization. If in these three things we have two things, we do not have the three things, then it is not called randomization. Then it's called the Cauchy control study. Is it okay? My dear students, it's okay. What are the important things?
Yes, sir. It's okay till now.
Yes, the important thing for the randomized control are three things are important. If one is missing, then it will not label, it cannot label the randomized control study, randomization. We have defined what is randomization, what is control, what is comparative control, what is intervention. Another thing is a high quality clinical trial is typically blinded. Unblinded study is usually not accepted, not good study, because in this, there are so many biased can be done. There are single blinded, the participants who are taking in part do not know about the medications, but the investigator knows. Suppose I am going to inject the vaccine. I know this is the vaccine, but I am not telling. This is no doubt unethical. It is unethical. But in certain cases, in very rare cases, the good study is that there should be, I think, there should be even I do not know about the medicine, or even other person do not know the person. And sometimes there's a triple blind study that the participant, investigator, the scientist do not know about the medication of this one. So this is blinded means this is the nurse that is doing the blind. This is the picture is showing blindness. There are three peoples. One is the patient, other is the investigator, and other is the scientist. So this is the one of the example of the, if you go on the Google, this is the study. There are so many studies which are double-blinded studies, randomized studies. So in the randomizations, there are certain phases. Phase 1, phase 2, phase 3. You have to understand about this phase. Because during last pandemic, the people were hesitating about this vaccine, that there is no randomization, there is no phase 3, phase 4 studies, and even in some countries, they started their vaccination, and even in Pakistan, the phase 3 studies, Pfizer and Moderna, and even other vaccines started in phase 3 studies. So I will give the detail. What is the phase 0? Duration 1 week, just a pilot study, participant 10 to 15, and the drug, very limited dose of this one. Choose determination of preliminary parameters, and if the medication acts differently. So in these pictures, this is the 0. Sometime in our thesis, we do the preclinical studies. In the thesis, in the dissertation, that is not phase, that is a preclinical study. And in this 0, primary goal is, what is the primary goals? Often skipped the phase 1, but the duration is very much. How many people take part in this one? So the durations of in the phase 1, duration is increased for 1 month. In phase 0, it was 1 week. In phase 2, it is 1 month or maybe several months. In phase 0, the participant was 10 to 15. And in this phase 1, the number of the participants. And why we do? To determine the determination of the safety, tolerability, and the other pharmacokinetics things and pharmacodynamics things. The main aim of this phase is to determination. So in phase 1 is continued, then the data is collected, but we do not submit it to the FDA. And in phase 3 is once again divided, phase 3A and phase 3B. These are the 2 studies. In the phase 3, the number of in phase 3, if you go phase 3, the number of the patients are about 1,000 to 2,000. So the number of the patients and even the dose, we are going to change it. This is single ascending dose or phase 1, that is a single number of the drug is given to small groups 3, and if they are safe, and we will increase the dose of after 3 members, then in a multiple doses we can calculate the safety. This is very important thing, that if the results are good, if the safety level is good and the dose is tolerable, mostly you can send this to the market. In the current pandemic, all over the world, the vaccine was started, and some of the antiviral drugs and some of the other drugs which were used in this, even steroids were used without going in the stage 4 because it was emergency. So it is sometime recommended that the FDA approves the safety level when you successfully conduct the phase 3. So this another thing is this, these are the different reasons of this one. So the phase 4 is actually the important phase. After that, it can give the properly commercial name, and even in first time the Pfizer and Moderna has given the different names after completing the phase 4. So this is the difference between the randomization and the non-randomized quasi-control. One of the sample is this. So these were the basic fundamentals, definitions, requirements of the interventional experimental randomized study. Another is called the cross-sectional study. The cross-sectional study, as in hierarchy, I told you it is analytical study, but it is observational. It is not interventional study. Cross-section means, suppose you are going to cook some good food and you want to take a taste of this. You want to check the salt level, or you check some other thing. You take a very small things with the spoon and you test it. That is called the cross-sectional study. The cross-sectional study is an observational study that involves the analysis of the data collected from the population at one specific point of time. They may be used to describe some features of the population, such as prevalence of an illness, or they may support conclusion or of cause of the effects. In this cross-sectional, I have used the terminology prevalence. Can anybody define what is the prevalence? Can anybody define what is the prevalence?
Assalamu alaikum, sir. It is number of new and old cases in the population of the disease.
New and old cases, total cases. Okay. What is the incidence?
Number of new cases.
New cases. One of the good example of this, suppose in first year MBBS, you test the 100 students for the tuberculosis, and you find then in the first year in 2020, there was one student suffering from tuberculosis. So the percentage, the incidence is 1%. Okay. At the end of the 5 year, if you in the final same class in the 5 year, in the prevalence, if you include the first year students, incidents and incidents during second year, third year and fourth year and final year, this will be the total thing, that is the prevalence. So this is the thing in the study, cross-sectional study. In the cross-sectional, simply you see the percentage of this one. So this is the example how you calculate the prevalence number of 6,139 people, 590 currently suffered from the incontinence and so had the condition of the particular time of the patient. Thus, the prevalence is total this one and calculated this one. This is the prevalence. This is the prevalence quantifies the proportion of the individual in a population who have the disease as a specific instance and provides an estimate of the mortality risk that an individual, actually the prevalence is going to show you the risk factor that the MBBS students face in 5 years from the first year to the final year. This is the formula. This is very simple. This is very simple to calculate this one. This is the incidence that we have. Another important study that is called cohort study. To understand what is the cohort study, this is a longitudinal study. Some people say cohort is an observational study, use investigation, the cause, the disease, the establish the link between the risk factors and the healthy outcome. Very well. The risk factor and the healthy outcome. If you go back, this is the cohort. Exposure, there is exposure and you want to see the outcome. In case control, there is outcome and you want to see the, these are two important things to understand between this one. So this is cohort study. What are the limitations of the cohort study? Expensive because it takes time to run and usually take many years to produce results. Unsuitable for medical students doing the research during thesis or during, because the students have very limited time. And rare diseases are there, needs large number of peoples. Offer clue rather than the defensive proof of this one. These are certain limitations of, these are the examples of the cohort study. The nurses' health study was established in 1976. When a cohort of 700 US female assigned nurses, these are our nurses, the age was 30 to 50 years, completed a questionnaire on medical condition and life study. The study was started in 1976. A total of 1,799 newly diagnosed breast cancer cases occurred during the first 10 years for a follow-up of from 1976 to the mid of the 1986, almost this year. Analysis were then conducted to investigate the relationship between the oral contraceptive use of and the risk of the breast cancers. Women who reported in the initial questionnaire in 1976 and in subsequent ones to have never taken oral contraceptives were considered to an unexposed group. In this, there are two groups. One is the exposed group and other is the unexposed groups. So keeping in mind these two groups, we have to calculate the inferential from this study. So this is that study that was published. That was published, and this is the. So in this study, we have to calculate the relative risk. In the first study, there was exposed and unexposed from the oral contraceptive drugs. So exposed cases and control. Suppose A was case and B was non-case. The total is A plus B. The exposed are A plus B in that parallel times. Unexposed case is the C and the non-case is the D. So if we want to calculate the relative risk, exposed divided by non-exposed. I'm putting that another example. Suppose risk, this is another example of this. Risk in healthy people for the 10 years, lung cancer and no lung cancer, then you control. With exposure to heavy smokers, there are 60 cases, and no, there was a 40 cases. The total is 100. Without exposure, non-smoker 15 developed the lung cancer, and 85, they were non-smoker. That's the 100. On this side, the calculation, the lung cancer was 75, and the no lung cancer, there is the 125. Can anybody calculate the relative risk? For 2 minutes to all, please do the calculation and please do the calculation and let me know. Over to you, students. This is the formula. This is the assignment for you.
I think it's 4. Sir, I think it's 4.
What is the number? What is the relative risk?
4.
So 4.
Okay, 4. Anybody
4.
else? 4.
So I
Anybody
think
else?
4.
So this is the answer. Good. Good. Excellent. Excellent exercise and excellent. So in this study, you are for if there are certain limitations that at what level it is important. I'm giving you another study, another example.
सर एक और सवाल है इसमें.
Let me know what is this question.
सर अगर फर्ज करें कि ये 60 नीचे आ जाता अनएक्सपोजर में और स्मोकर्स में वो कम हो जाते, 15 लोगों को सीए हुआ होता, तो ये
Sorry sir, can you in English, because this is your answer.
Okay, sorry.
Your answer is here.
Yes.
Your answer is here in this question. If the relative risk, if the relative risk is lesser than 1, means the event is less likely to occur in the experimental. Yani then it is less likely to the experimental group. If the relative risk is greater than 1, means the event is more likely to occur in the experiment. Now it is clear? So the relative risk is relative. And if in this question, with exposure there are 15 and without exposure there are 60, again we have to calculate the relative risk. We want to see the exposure and the outcome. If the relative risk is lesser than there is no association with exposure to the outcome. And if it is greater than 1, then there is a definite association with the risk to the outcome. So this was about the cohort study. Okay, another, can you calculate the relative risk of this study? There are total 5 people.
So 1.6.
Okay, good. This is the 2.7. Can you see it again? Can you check it again? This, can you check it again?
Okay, sir.
The formula was this. I gave the pre-exposed divided by this. This is the A is over by A plus B, C is C plus D multiply. So if you apply same here, this is an example of this one. 145 by 18 is 0.77. 89.312 is equal to 2.2. So this 0.77 divided. So in this way, association is not stronger. No doubt it is greater than 1 as compared to our previous study. So these are the different fundamental things that which things in which study, which things are required to draw the inferentials. In observational study, when it is descriptive, then there is no need to draw something. There is no need to make an hypothesis. When it is cohort, you have to see only the prevalence and the incidences. When it is cohort study, you have to check so many other things, relationships and the relative thing. There may be so many other things you can check, but very important thing is that exposure and the risk. So I'm going to start this study. There are so many drawbacks, there are so many limitations, but my dear students, my advice is to make a very good questionnaire to reduce the biasness. Okay, another study is, so the same thing, when is there exposure to check the cohort, discard the outcome, this is the summary, when there's outcome, this is cohort, this is called the case control study, this cross-section study, time out one. Another is the case control study. You can do even the students in their laboratories, the students in their pictures, in their students, in their thesis, in their research, they can do the case control study. But it is very important to check what is the case and what is the control. A case control study is a type of observational study in which two existing groups differing in outcome are identified and compared on the basis of some supposed causal attributes. The case control studies are often used to identify the factors that may contribute to the medical conditions by comparing subjects who have that conditions or disease, that is called the case, and they are not healthy. The patients who do not have the condition or disease, but they otherwise are similar, they are living in that area. I am going back. Now there is outcome. Suppose there is a cancer. I want to see that what is the exposure, why the cancer is happening. So in this way, we will search the people who are there and their condition, they're suffering with the condition of disease. And similar people will check with or without disease for that things. Example, a case control study was carried out to assess the relationship between the cervical cancer and the exposure to the human papillomavirus 16 or 18, smoking, and selected. You can even check two exposures, smoking and one in this, the one human papillomavirus and the smoking, and selected aspects of the sexual and reproductive behavior. There are so many other things you can check in this study. The study included 436 incident cases of the cancer, cervical cancers, and 387 controls of similar age randomized selected from the gender population. And these are the two populations that generated the case of this one. Suppose this is the study that, and you can check from the Google. So what is the advantage? It is relatively quick and easy. Easy, does not need large number of the people, suitable for identifying the cause of rare disease or sudden breakouts, limitations, recall bias, sampling sometime maybe not. So a case control study identifies cases of the diseases first and then analyze exposure to the risk factors, whereas the cohort squad study is opposite, following the exposure data and watch any emergency of the cases of the diseases on these two sides. You are showing these, the case control study and the on the right side and the cohort study. And in cohort study, we checked the relative risk, but in this we have to check the odd ratio. Now I'm going to another slide giving you the assignment. What is the odd ratio? It is one of the range of statistics used to assess the risk of a particular outcome if a certain factor's exposure is present. It is a relatively measure of the risk, telling us how much more likely it is someone who is exposed to factor under the study will develop the outcome as compared to someone who is not exposed, mean control and the case. So this inferential is used in the case control study. Interpretation: if the odd ratio is clearly greater than 1, then the conclusion is those with the disease are more likely to have been exposed, and there is no more association. Whereas if it is close to 1, then exposure and disease are not likely to be associated. And if it is less than 1, then this just the exposure is a protective. A similar, you asked the question when we are doing the cohort study. So the number of 1 is very important. If it's greater than 1, then there is exposure. And if it is equal, then the exposure and the disease are not likely to be associated. And it is, if less than this, the exposure is then protective. This study was usually done when we were doing the study in King Edward Medical University once, and we saw that in some of the cases, the aspirin was protective. We're doing for the heart disease, but in stomach, in another cases, this was, we are doing the study in for the gastric ulcer, but we saw that this was protective for the heart diseases too. So odd ratio, how to calculate the odd ratio? This is the odd ratio. This is the example. Can you calculate the odd ratio from this one? There are two groups, lung cancer cases and no lung cancer cases. Heavy smokers, there are 283 people who developed the cancers and no lung cancer in this. They were also heavy smokers, 7 to 80, and non-smokers 64 developed, and in non-smoker 1010D. So in this calculation, what is your result? What is your answer? Anybody?
6.1.
It is
6.1.
6.1. Okay.
Yes, 6.1.
Excellent. Excellent. So this is another case. Can you calculate the odds ratio? Heart disease about this. Study was carried out to determine whether the tonsillectomy was associated with subsequent development of the Hodgkin's disease. 109 cases of the Hodgkin disease were identified. 109 controls without diseases, but with tonsillitis. So what is your result?
So 2.86.
2.8.
Okay, what it means? What is your what it is showing? What you mean by
So
2.8?
it is more than 1. So there is
No,
association.
but why either the tonsillectomy was associated with Hodgkin?
2.86 is more than 1, so it is associated.
Yes, good. Yes, good. Good. So this is 2.8.
Yes, sir,
It
2.8.
mean- 2.8, which means this is your hypothesis was correct. Here, this is this is the study of this one. So we have covered so many fundamentals of our studies that we usually have to do during our research. Another thing sometimes people do call this is the primary research or this is the secondary research. You should know what is the primary research and what is the secondary research. Primary research means it is defined as a factual first-hand accounts of the study written by a persons who have a part of study. A primary research tends to come first in publication cycle. Primary research can be conference paper, article published in the journal or in the thesis. While the secondary research, it is defined as analysis or interpretation of the primary research. It tends to come a second in publication. The method of writing secondary research is to collect primary research that is relevant to a writing topic and interpretation. What a primary research found, secondary research can be a review or meta-analysis. Can you give me the example of primary research and secondary research? Suppose there are two peoples, one is Zaid and other is a Bakr. The Zaid has completed the PhD on any topic of the breast cancer. He has seen or she has seen the association of breast cancer with contraceptives, and he has published his result. This is the primary research. Suppose Zaid has not done any research and he wants to do the research. He will collect the data from this primary research, not only this research, he will collect the data of similar research from others, and then he will do the research and then he will do the analysis, meta-analysis. This does not mean this is not original research. This is original research, but this is the secondary research. So hierarchy of the research designs to level of scientist. This is from similar you have studied in medical education. What is the primary research and that is secondary research. In the primary research, you do it yourself. Either it is observational or either it is a cohort study or either it is an interventional study and you publish the paper. But after 4 years, 7 years, anybody want to make something, some new things or make to do some new results, he collect the data and compare the data. This collection of data is called review. That is called secondary review, systemic review. And when from systemic review, you will draw the inference, that is called the meta-analysis. I will give the example of this one. What is that is called systemic review. Systemic review is a type of literature review. There are so many reviews. Systemic review and literature review, two are different things. Literature review is only to give the idea about your study, while the systemic result is giving you the quantification, giving you the quantitative analysis of their different studies. These are two different. So systemic review is better when you are going to write an article, while the literature review is better when you are writing your research or dissertation or your thesis. The researcher use organized method of locating, assembling, and evaluating a body of literature on a particular topic using a set of specific criteria. Preferred reporting item for systemic review and meta-analysis, that is called PRISM. P, preferred. R, reporting items for analysis, systematic, and R, meta-analysis. So the systematic review may also include a quantitative pooling of the data, and the calculation is called analysis. Once, if you will simply review it, then it's called systemic review. But if you calculate the if you will quantify it from the pooling, the already done the primary research, then it is called the meta-analysis. So this is the example of type of review, systemic review. Individual patients on this was a review that are not systemic, and sometimes they draw. So the students can write and research paper, original research paper, if they do not have the primary research, but it is very difficult to calculate the results from the other study, that is called the secondary study. Preferred reporting items for the systematic review and the meta-analysis. So this is the table that how you will collect it, how you will record it, a recording calculation of this method. So this is something, it's very cumbersome, this type of the research. Meta-analysis, as I told you, the difference between this one's calculations. Different study that use of this you can use for the systematic review and the meta-analysis. Different websites you can use. I have given these websites. You can even calculate from the PubMeds. Usually I do mostly from the PubMeds. There are so many websites that the people use. Practice guidelines for this meta-analysis. A statement produced by a panel of experts that outline the current best practice to inform healthcare professionals and patients in making clinical decisions. The statement is produced after an extensive review of the literature and is typically created by professional associated governments, agencies, NGOs, your different institutions you can choose. A good line clearly defines the topics. Topic is very important to select it. Appraise and summarize best evidence regarding the prevention, diagnosis, prognosis, therapy, harm, cost, anything you want to do in your systematic review and analysis. So what is the advantages of this? This is secondary, no doubt it is secondary research, but there is what are the advantage created by the panel of experts. Even a single person is very difficult to do. based on the professional published literature already. Practical guidelines for the clinicians considered as evidence-based resources. What is the difference? Slow to change and update because you have to collect the data from so many studies. Not always available. Sometimes literature is very deficient. Data is very deficient. So sometimes it's expensive and time consuming. So this is one of the example of the systematic review of this. This is an endometrial infertility. So this was all the different basic fundamentals of the research. The summary of my today's lectures is that you understand first are different definitions of the research and make good study designs. And if you are going to make the study designs, so this is the summary of today's lectures. What is the basic fundamentals of your research? And what are the basic things that you want to draft the summary from this one? Any questions? Can you revise? What is the odd ratio?
So odd ratio is हमने जो calculate A into D divided by B into C.
Yes. In which type of study we use the odd ratio?
It's a case control study.
Yes, good. What is the case control study? What is the case? How you will define the case and what is the control?
So when there is an outcome and
If there is,
a
yes,
outcome and we
yes,
go to
good.
calculate the risk.
I told you people that it's not important that there may be equal numbers, but the condition may be same. They are living in a similar situations. Okay, Dr. Fazila, can you answer me? What is the relative risk? What is the relative risk?
The relative risk is A plus B divided by C plus D.
Okay. Okay. In which study we have to use the relative risk?
The cohort study.
Okay. And what is the importance of this? And what is the importance parameter in this relative risk? How much is important?
Sir,
One, if parameter is relative risk 1. Okay, Dr. Bhomik, you are there. Can anybody define what is the incidence?
Okay, incidence is the new cases.
Okay, and the prevalence mean?
Prevalence basically both the new and old cases.
Okay, thank you. Dr. Khalid, can you define what is the systematic review?
A systematic review is that you gather a lot of studies and analyze their data and form a comprehensive, article. And this is considered as a secondary data.
Thank you so much, Professor Khalid Chima Saab. Okay.
Can we add, it actually collect and critically analyze multiple research study.
Yes,
The critical analysis is important.
Okay, collection of the is very simple. That collection of the primary studies and doing the critical analysis. And then what is the meta-analysis? What do you mean by the Dr. Atka? What is meta-analysis?
So meta-analysis, when we do, it is a secondary research. When we collect the data or from the other similar researches, and then we do the analysis of their results, critical analysis.
Thank you so much. I will share my different questions. I will share my MCQs on
Sir, I
your
have a question regarding meta-analysis.
pardon?
Sir, I have a question regarding meta-analysis.
Yes.
Like if I want to study any relation between, like I want to do a meta-analysis, is there a certain number of studies I need to take, or
Yes,
a
you have to
certain
take. Yes, you have to suppose there is one study that was done by Dr. Bukhari. That was the association of contraceptive with breast cancer. And I have drawn an inference that if you want to do the meta-analysis, you will collect the similar study from other authors, and you will compare the data, collect the data, and then make the summary. Suppose there are 10 authors have done the study on 10,000 patients, and if they say that there is some relation of oral contraceptive with the breast cancer, then this is called, if you are doing only critical analysis, then it is systematic review. But if you are going to draw that in 10 studies, you want to make the clear question that either these contraceptives are really associated with the breast cancer, then this is called meta-analysis.
So my question is, how many studies will I take if I intend to do the meta-analysis?
I
Is
think
there a number?
greater, I think, Dr. Atka, greater the study, the better will be the results, and better is called the meta-analysis.
Okay, sir.
It should not be less than 10 studies.
Okay, sir. Thank you.
But if you have more than this study, greater will be the study, greater number of the studies, the better will be your systematic critical analysis, and better will be your results. Any other questions?